Okay, folks, so this is part 2 of our chapter 15 discussion of the innate immune system. So, what we've talked about so far are just the barriers that our body puts up to prevent potential infection or invasion of foreign particles into the body. Predominantly, that starts off with the physical barrier, which is our skin. But then we also layered in a discussion of our sweat and tears with various chemical factors that help prevent infection and invasion of pathogens, as well as our discussion of mucus, which has both chemical, abiotic, and biotic factors that are actually helping to provide that barrier and clearance. Now we're going to start our discussion of the immune system and the cells of the immune system. And a great place to start is the blood. Although the cells of the immune system occur throughout the body, we've already met our dendritic cells and our mast cells, but the blood really provides the highway that connects all the different signals between and the cells themselves as they move through the body, as well as the proteins that complement or work with those cells. So the blood is a great place to start. And our discussion of blood will also lead us to a discussion of pus. So stay tuned. Okay. So beyond the obvious of like what is blood, what is in blood? So predominantly blood is water, but then dissolved within the water, and you can see here this image has blood unseparated and then separated, where you can see the liquid component and then you can see the cells kind of settled out there. Within the liquid component, there are electrolytes, ions, nutrients, a number of proteins. So you have proteins that facilitate gas exchange, you have proteins that facilitate the exchange of nutrients, you have proteins that actually complement or work in parallel with the immune system. So a number of really important proteins that are within blood. Also, you have antibodies, which are an important part of the adaptive immune system that are carried throughout the body in blood. So that's that plasma component. If you remove these various components of plasma, we just call it serum. That liquid base for blood, we call it serum. Okay, then we also have the living components of blood, which are erythrocytes, which are red blood cells, and our leukocytes, which are white blood cells. Okay. And so, considering the living components, we call these the formed elements of blood. Our erythrocytes predominantly are tasked with gas transfer. Platelets are previously living cells that were broken up into components, into various subunits, and they help with blood clotting, which is really essential to the immune system because the skin is the barrier, sort of the walls of the fortress, right? And if there's an abrasion in that barrier, then it allows for a continued reintroduction of pathogens or invaders. And so platelets are really important to preventing pathogens from entering when there is some sort of invasion in or abrasion in the skin that allows for invasion of pathogens. Leukocytes, though, are a little even more active. They're white blood cells that have various tasks in identifying pathogens, actually directly killing pathogens, and also amplifying the response throughout the immune system so that that pathogen, that invasion can be dealt with and then remembered over time. And that's the adaptive immunity part, which we'll get to in chapter 16. So let's meet some of the cells of the immune system. Okay, so we have this diagram here, which is kind of a really great foundation for the discussion that we'll have in this portion of the lecture. So maybe it's a great diagram to take notes on, but we'll also continue to go through this in the upcoming slides. So if you look at the cells of blood, you'll see that in terms of the origin, there are 3 stem cell origins for the cells of blood. One, as our erythrocytes it results in our erythrocytes. Then we have these myeloid stem cells that result in our platelets, which we talked about being really important for clotting. And then basophil cells, neutrophil cells, and eosinophil cells. Okay. Then we also have monocytes that come from the same myeloid cells. And then we have this third branch or ancestors for these stem cells called lymphoids or lymphocytes. And these result in three types of cells. One are natural killer cells, and then the second and third are TMB cells of the adaptive immune system. Actually, let's go ahead and add those in because that helps us kind of remember. So, natural killer cells. Okay. And B lymphocytes and T lymphocytes. Okay. So those three types of cells will result from a lymphocyte. Okay. Okay. So let's learn a little bit more about each of these. So, in terms of the function of the immune system needs to have patrols that are looking for pathogens. It needs to have amplifiers that are sending out messages about pathogens. It needs to have cells that come in and take out pathogens. So they're very effective at just killing pathogens. And it also needs to have cells that can remember pathogens. Similarly, it needs to be able to do the same thing for cells of our own body that either are infected with a virus or are cancerous, right? And so we'll go through these different cells and talk a little bit about their function in all of those. Okay. So, in terms of recognizing pathogens, we've actually already talked about one of the most important cells for recognizing the initial invasion of a pathogen. Who remembers that? The very top layer of skin, the network of C-star shaped cells that we talked about in part one. Okay, so I'm hoping that you said dendritic cells. Dendritic cells just hang out within our skin, and they actually provide a network that allows us to initially just kind of recognize pathogens and phagocytize them, so actually engulf and break them down. And then there's also another group of cells that do a very similar task. They're called monocytes. They go through and peruse and break down potential invaders. They also, when there's no pathogens around, they just kind of collect garbage, so dead skin cells and things like that. And the difference between big difference between dendritic cells and monocytes is that dendritic cells, they will actually eventually migrate out of the tissue and to lymph nodes to actually bring information to our adaptive immune system, whereas monocytes tend to stay within the tissue and collect and get rid of pathogens. Well, I don't see I don't see macrophages and I don't see dendritic cells on here. Well, that's because both of those types of cells come from this ancestor right here, monocytes. Okay, so let's add those in. Okay, so right here under monocytes, maybe I'll put it above monocytes actually, because there's some other things that we need right under monocytes. Okay. So right above here, we've got our dendritic cells and our macrophages. And both of these originally, so they are coming from monocytes, both of these originally hang out in the tissue and just literally degrade potential pathogens that come out. But remember, dendritic cells also send that information to the immune system as well. Okay. So those are kind of initial within the tissue. Also, we said that there was another type of cells hanging out in the tissue. and that was mast cells. Do you remember when we said mast cells were hanging out in the second layer of skin, and they were just amplifying the immune system kind of call out or immune system alert when a potential pathogen was invading. And so you also don't see mast cells on this particular diagram, unfortunately, but mast cells in the tissue are very similar to basophil cells in the blood. So I'm just going to put mast cells right here, mast cells above basophil cells right there. Okay. Because basophil cells do the same thing, but they're in the blood. And they literally, when you have your macrophages or dendritic cells calling out, okay, I found a pathogen and I'm phagocytizing it, I'm devouring it, then they'll actually call out with various types of chemicals like cytokines, and then the mast cells and the basophil cells, mast cells in the tissue, basophil cells in the blood, will amplify that by continuing to release chemicals like cytokines. They're kind of like the siren of the immune system. Okay. Then when you call out, what happens? Reinforcements come, right? So macrophages are one type of reinforcement, but the big reinforcement, and these are actually the predominant, like in terms of quantity, in terms of numbers of white blood cells, the most, uh, um, the most in terms of quantity are neutrophil cells, these guys right here. And these guys come in, they engulf and phagocytize the pathogens within those areas, and then they die in mass. These are our foot soldiers, our neutrophil cells right here. Okay, so we can add maybe a note, or I'll let you add that to your own diagram. So I'll just keep just the names of the cells on here. Okay, so your neutrophil cells come in mass. They come in as these mast cells and basophil cells are calling out for reinforcements, and neutrophil cells are also called pus cells. Neutrophil cells are called pus cells. And the reason why they're called pus cells is they're the ones that come in such big numbers, but they also are very short-lived. And so they phagocytize, they try to take out the invader, but they die, and they die in mass, and that's where you get that white buildup of pus within a pimple or a cyst or something like that. And in a minute, we'll watch a video about pus as well. Okay, so that's our basophil cells, that's our neutrophil cells. Eosinophils are similar to some of the other cells in that they're part of the immune system and they're important to taking out various pathogens, but they tend to specialize a little bit. And they are phagocytic, so they can engulf bacteria or smaller pathogens, but they actually specialize in large pathogens, particularly parasitic eukaryotic worms. And so they also have the tools to kill cells from the outside, because just one white blood cell is not going to be able to engulf a whole like large parasitic eukaryotic worm. It's going to be too big. The worm itself is going to be multicellular, and you're talking about just one single white blood cell. Um, so instead, it also, not instead, but in addition to being able to phagocytose small bacteria, it also can just puncture holes into larger worms. And so it has basically two killing mechanisms. And we'll get back to that in a minute, but we're just kind of doing a survey of the cells that we see here. Okay. Then we said our monocytes, those tend to evolve into or mature into dendritic cells and macrophages, both of which hang out in the tissue and provide that initial kind of recognition of potential issues. Then our lymphoid or our lymphocyte cells are pretty unique in that First of all, B and T cells are related to our adaptive immune system. And so we're going to talk more about B and T cells in chapter 16. But our natural killer cells are also unique. So what we've talked about before are cells that are looking for extracellular or extra organismal invaders like pathogens, bacteria, parasitic worms, maybe viruses. But our natural killer cells are on the lookout for our own cells that have some sort of issue. Now, that issue could be a viral infection, right? And so they're not acting normally because of a viral infection, which is actually a pathogen or an invasion, right? But it also could be cancerous cells. So our natural killer cells are looking for cells of our own body, and it will actually neutralize them by puncturing them and with a chemical that will essentially lead to apoptosis of those cells. So our natural killer cells are more unique to looking for cells of our own body that are not functioning correctly rather than specifically looking for pathogens. But do remember that sometimes those cells they are infected, and that's why they need to be eliminated or removed. And then our B and T cells allow us to create long-term memories, antibodies, and responses to potential pathogens as well. So in that way, we can look at all of these different cells and get an idea of kind of the progression of how our various armies come together to protect us against pathogens, and then remember how to protect us later on, which we'll talk about more in chapter 16 when we talk about adaptive immunity. And it also gives us an overview that's a little bit more easy to process in terms of the function of these various cells, because I think your book kind of focuses on classification, which is important, but doesn't always allow us to really easily understand the function of each of these types of cells. Speaking of classification, let's mention some of that. So within this diagram, you can see our basophil and mast cells. Their primary goal is to recruit and signal for the immune system to respond. So for example, inflammation and calling out. Whereas you have some cells that are capable of phagocytosis. And then we also have this distinction, granulocytes and agranulocytes, okay, which we can talk a little bit more about now. So granulocytes essentially are cells that have these granulocytes within them, which are compartments or granules that have a number of different chemicals and enzymes that allow them to phagocytose or kill cells. And so our basal cells, eosinophils, neutrophils, they have these compartments. Now remember, basophils are not predominantly phagocytosing. That's not their predominant task. They're calling out for help by releasing chemical warnings, but they are capable of phagocytosis. And then eosinophils, we said they're capable of phagocytosis, but they also have the capacity to actually inject some of those chemicals into a parasite that's too large to actually engulf. And then neutrophils, we said, are basically professional at phagocytosis. They just come in and engulf pathogens. And we'll talk more about how the process of phagocytosis happens. But as we're talking about it now, just know that that's the white blood cell engulfing the pathogen and breaking it down, engulfing the pathogen and breaking it down. And neutrophils are particularly professional at this, although they come in mass and they die quickly. They come in mass and they die quickly. So neutrophils are our pus cells. Okay. And then, so remember, we said basophil cells can technically phagocytose, but it's not their predominant task. Okay, so this is kind of like both and. Okay, then our agranulocytes are the cells that tend to lack an abundance of those different enzymes and chemicals, although many of them still have some of those enzymes and some of those chemicals that allow them to fight off pathogens directly, but that's not their predominant task. Okay. And monocytes, they are still phagocytic, even if they're not, they are agranulocytes. So they still do have some of those granules that allow them to be phagocytic. But to be honest, the distinction of granulocyte and agranulocyte is mostly based on how cells show up on the microscope. And so some of those granulocytes are more visible on the microscope, even though something like a macrophage or a dendritic cell might have many of those same structures, but at a lesser so, and then they don't show up on the microscope quite as much, quite as easily. And so this distinction is a little bit less functional than it seems like in the text, in the reading. Okay, so in terms of agranulocytes, we said we have our monocytes. Our monocytes are our dendritic cells and our macrophages. So our macrophages, we said they are, let me move this out of the way so we can see. They're just resting, perusing, looking through the tissue, and they collect garbage. So dead skin cells and things like that. And then also when they encounter a pathogen, then they can also do phagocytosis. Okay. And then dendritic cells, they also can do phagocytosis. But then once they do phagocytosis, they actually hold on to the components of that pathogen and they present those components to the immune system. And they don't do it in that specific cell tissue where they were. They actually migrate. They actually leave the skin tissue or they leave the tissue that they're at because there's different dendritic cells throughout our body. And they migrate to lymph nodes so that they can present. They're professional antigen presenting. They present those pieces of the pathogen that they found to the adaptive immune system, to B and T cells. And there is a little bit of a difference between how dendritic cells, different types of dendritic cells, like dendritic cells and follicular dendritic cells, and so on and so forth. We don't really need to know those distinctions in how they present the components of those pathogens to B or T cells, but just that it's just the fact that they pick up essentially from the battlegrounds information. They're kind of like informants and they take it back to the immune system is really amazing. Okay. And we said whenever they leave the tissue, their task is no longer phagocytosis. It's the professional presentation of antigens to the adaptive immune system. But before they leave, they do recruit more monocytes to turn into dendritic cells and take their place, right? So really amazing, really cool cells in our immune system, these dendritic cells. And then we also said we have these agranulocytes, which are lymphocytes, and those include three cell types. So our natural killer cells, our B cells, and our T cells. We said the natural killer cells, they are tasked with identifying cells of our own body, human cells, that are either infected with a virus or cancerous. Okay, so let's see. ID and eliminate infected or cancerous cells. Okay. And in that way, we can see this just amazing kind of this amazing symbiosis. It's not symbiosis because it's not different microorganisms or different species, but it's all of these different cell types that are working together to identify, eliminate, communicate, amplify, and remember the potential pathogens or foreign objects that come into our body. Okay. Um, so let's continue our discussion by mentioning that we can actually sometimes look at the different amounts of these different white blood cells and get an idea of what potential infection we might be having. So we already talked about eosinophils. Eosinophils if there's an uptick in white blood cells that are particularly eosinophils, then we might say, oh, maybe this is a eukaryotic infection or a parasitic worm. Um, Also, bacterial diseases tend to have, we see an uptick in leukocytes and neutrophils that come and answer that call. And then viral infections tend to see an increase in lymphocytes rather than some of these other cells. So it's not foolproof, but looking at what are the components or what are the kind of differential expression of these different white blood cells can give us an idea of what's going on. Okay, so we've been talking about these different white blood cells and their functions. One of the primary functions of many of these cells is phagocytosis. So yes, we have these cells that kind of identify these organisms and take them out. How do they take them out? Well, there's a number of different ways. We're going to talk about them, but one of the predominant ways is phagocytosis. So let's talk more about what phagocytosis is. Okay. Um, so many of you might already be familiar with this action, within the body, but even if not, if you know about an amoeba, right? An amoeba is this just amazing organism that's a single-celled predator in the environment. And I just think that that is just, wow, it blows the mind, right? So you've got this single-celled organism that can be a predator, and the way that it does that is it has these pseudopods, which are kind of like arms that extend out of its cellular envelope. And it just uses its cytoskeleton to move those arms. And then there's receptors on those arms that allow for it to kind of see or feel the different types of prey that it can then engulf. And then once it engulfs them, it actually puts a vesicle around that prey item, which is another single-celled organism in a pond, for example. Um, and then it just injects different types of enzymes and chemicals. So think back to granulocytes, right? And those granules within those granulocytes, and that digests that prey item, and then it can, take those nutrients in and feed itself. Now, a white blood cell, its goal is not necessarily to nourish itself, but actually take out a pathogen, but it's got that same idea. And so, since sometimes folks are already familiar with amoebas, I like to show this video of an amoeba, and then that'll allow us to talk about what phagocytosis looks like. Okay, and so essentially the amoeba is going to move and then recognize what it wants to take out and then start to ingest it and then actually take it over and eliminate, or in the case of the amoeba, ingest that prey item. Okay. Okay, so you can see the amoeba here. Oh, I won't put my cursor over it so you can see. But you can see the amoeba and its pseudopods, those arms are reaching out. This is just a single cell that's doing this, which is amazing. And then it's able to reach its arms around that little swimming single-celled organism that's in pond water. And eventually it'll enclose that whole space 3D around that prey item, and then it'll release enzymes and chemicals into that vesicle and actually digest it. And so, since sometimes we're familiar with this concept with amoebas, then we can go back and say, okay, well, the steps of phagocytosis that an amoeba takes, similar to the steps of phagocytosis that a white blood cell takes to take out an invading pathogen, are as such. There's chemotaxis, so the movement towards the thing, right? And so, how does it know to move towards whatever it's going to phagocytize? Well, the amoeba has receptors on the outside of its cell that say, oh, Okay, there's molecules in that area that seem to be similar to the molecules that are released from a prey. How about in the immune system? Well, white blood cells look for something called PAMPs, and we'll talk about those in a little bit. But these are essentially just the traditional signs of a bacteria. Okay, it seems to be releasing these types of molecules that bacteria tend to release. It seems to have flagella and fimbriae. Well, human cells don't have flagella and fimbriae. Okay, so it seems to have, peptidoglycan. Well, human cells don't have peptidoglycan. So these general kind of ideas or general components of a bacterial cell or an invader, they can actually recognize that and say, uh-oh, this looks like it might be a pathogen and engulf it. Well, before they engulf it, but move towards it, right? Move towards it, then actually adhere to it and then form a vesicle around it and then digest it. Now, sometimes these professional antigen presenting cells, like dendritic cells, will then display what they've digested on the outside of their cell. And that's how they present those parts of the pathogen to the adaptive immune system cells, the B and T cells. So, in these really gruesome like stories of historic wars where you have a warrior like wearing the finger bones or the rib bones, or even, folks displaying a skull of whatever, enemy soldier, is very similar. That cell is literally presenting the parts of the pathogen on its external cell membrane, on its external cell, so that it can show it to the other cells, the adaptive cells of the immune system. So, very interesting. Not all immune cells do that professionally, but we talked about dendritic cells as one example. Okay, so here are just two videos on phagocytosis.
Phagocytosis is the ingestion of a solid, such as a microorganism or cellular debris, by a eukaryotic cell. Phagocytosis is used by some phagocytosis for obtaining nutrients, but this process is also used by certain cells of the immune system to fight infection. These immune system cells include macrophages and neutrophils and are collectively called phagocytes.
Okay. And then here's the process for you.
Chemotaxis is the movement of a cell toward or away from a chemical stimulus. Phagocytes use pseudopods to move toward microorganisms or damaged cells at the site of infection, often arriving only minutes after infection. Chemotactic chemicals that attract phagocytes include microbial products, components of damaged cells, chemicals released by other white blood cells, and peptides derived from the complement system. The plasma membrane of phagocytes usually attaches to glycoproteins on the microorganism's surface. This process is called adherence. Adherence is made easier through opsonization, a process in which antibodies or complement proteins from the host coat the microbe's surface, serving as handles so that the phagocyte can more easily attach to the microorganism. The proteins that coat the microbe are called opsonins. The pseudopods fuse, forming a sac called a phagosome. The phagosome enters the cytoplasm, where it fuses with lysosomes that contain digestive enzymes and other antimicrobial substances. The fused structure is called a phagolysosome. Digestion of most bacteria is complete within 10 to 30 minutes. After phagocytosis, the remaining debris is eliminated from the cell by exocytosis, in which the phagolysosome fuses with the plasma membrane and expels its contents. Some of the contents of the phagolysosome may also be presented on the cell membrane through a process called antigen processing and presentation. Let's review the steps in phagocytosis. The pseudopods of a phagocyte use chemotaxis to approach the microorganism. The pseudopods adhere to opsonins that are attached to the phagocyte. The microbe is ingested within a phagosome. The phagosome fuses with a lysosome to form a phagolysosome, in which digestive enzymes break apart the microorganism. Finally, the remaining debris is eliminated by exocytosis. Parts of the microorganism are presented on the surface of the phagosome.
Okay. Okay. So we know that that these white blood cells can do phagocytosis, but we know that some of them also do other things. So let's just go through and talk about how each of the white blood cells does its task. So killing by eosinophils, we said that the eosinophil can actually attack the eukaryotic worm, in this case called helminths, just a specific class of eukaryotic worms, by actually adhering to their surface. The white blood cell, the eosinophil, will adhere to its surface, and sometimes it will actually have help from antibodies that actually will aggregate the worm, but also the white blood cell, and then it can secrete toxins right onto the worm itself, and that can help to break it down. Okay. Then, in terms of neutrophils being pus cells, they are the most abundant and short-lived. They can destroy the microorganism through phagocytosis, um, but they also have the chemicals to destroy them elsewise. So they can also release chemicals that will kill nearby pathogens. So one of the reasons why neutrophils are so short-lived is because they're so effective. Like they are the kind of kamikaze soldier of the immune system, and they're really really effective. And if they stay around too long, they can actually start to cause tissue damage. And so it's really important that you get neutrophils in mass to the site, but then they finish their task really quickly, or otherwise you can have lasting tissue damage in that area. Okay. Um, and that's what we just did with neutrophils. Then moving down in that diagram there to our natural killer cells, we said that instead of phagocytosis, because our natural killer cells are not really looking for small invaders that they can just engulf, they're actually looking for cancerous or infected cells, host cells. And in that way, they actually will secrete toxins onto the surface of the cell and then kill it that way. Okay, so we know that these different cells can either phagocytose or do various types of chemical killing of an invader or a cell that is infected or tumors. But how do they recognize invaders? That's a really important question. They need to know what they're actually going to phagocytose. And we had mentioned this briefly earlier in this lecture, that essentially there are receptors on the cells that are looking generally. Now, when we get to chapter 16, we're going to talk about specific, like receptors that are looking for specific antibodies, and that is also true. Okay. But for this first line of initial defense, they are just receptors that are generally looking for patterns that are similar across different types of pathogens. So looking for, okay, peptidoglycan, different types of um, flagella, fimbriae, pili, things that usually the host cell wouldn't have. And those receptors either are in the membrane of your white blood cells or in the cytosol of your white blood cells. So you have these are called pattern recognition receptors. And the ones that are on the membrane of our white blood cells are called toll-like receptors. And the ones that are inside the cytoplasm or the cytosol, which makes sense because eventually it's going to be these pathogens are going to be engulfed through phagocytosis, are called nod proteins, which are kind of a long name, which is nucleotide binding oligomerization domain patterns. So it's kind of this long name, but essentially these nod proteins that are inside of the cytosol of the white blood cells. And so what they recognize are those patterns. And these patterns are called PAMPs, pathogen associated molecular patterns, which are just general patterns. Like if you were to go into a gathering and generally see someone who you didn't recognize, but not specifically know who they were, right? They kind of like act differently or look differently. Generally, you see someone who you don't recognize. And then the adaptive immune system might say, okay, I need to actually know who that person is very specifically. And so that general recognition of pattern associated molecular patterns that are on those pathogens allows for those receptors to bind to those PAMPs, those molecular patterns, and then take out that specific invader or lead to apoptosis if it's a host cell. Okay. Okie dokie. So that's basically our discussion of white blood cells and kind of their actions. So I would recommend, or I definitely recommend taking a break here. But in that break or before that break, let's watch the video on blood and the video on pus. And so the video on blood is a really great just overview of everything that we've talked about. And the video I'm using this time around for pus is actually a student generated term project video, which is very, very exciting. Um, and it focuses on a specific type of microorganism within the microbiome, but you can see that the response of the body would be similar across the board, regardless of the type of pathogen that would get into a follicle. And when the video is talking about the recruitment of white blood cells, it's specifically talking about neutrophils. So go ahead and watch those videos and then come back into the lecture and we'll continue. Okay, folks, welcome back. Um, so in this next part, we're going to go through and we're going to talk about some of the proteins that are active within and parallel to the immune system. So they allow for communication and response between the immune cells, and they allow for a body-wide response to infection. And we're going to talk a little bit about some of those body-wide responses. Okay, so we're going to talk about interferons and the complement system. Interferons are really interesting because remember, when we're talking about the immune system, we're talking about two different potential kind of dangers. One is the invasion of a pathogen from the outside, and one is a cell itself that's no longer functional. Either a host cell itself that's no longer functional, either because it's cancerous or because it is infected by an invader. And so interferons are specifically related to cells that have actually been infected by a virus. And as they're going down, so remember when we talked about chapter 13 and we talked about the infection of a host cell, and basically that virus takes over the replication machinery and forces the cell not to do its own functions, but instead to replicate the virus. As that host cell is going down, and as it's no longer able to save itself, essentially what it'll do is release these proteins or make these proteins called interferons. And interferons are this last call, not even for help, more of a warning to the adjacent cells, because those virions are going to be released from that infected host into the environment, and then those adjacent cells are the ones that are going to pick up that potential new viruses. And so rather than this being just a very localized, it actually spreads out through the tissue in that area and it can actually lead to a full body response. So some of that fever and fatigue and tiredness and muscle aches that come from viral infections are actually because of these interferon proteins that are being released into the environment around a cell. So what does that look like? Well, here you have a cell that's been infected by a virus, and the virus starts to take over. Well, the cell makes these interferon proteins and releases them into the environment. And that actually leads to the adjacent cells taking precautionary mechanisms. So, preventing entry of certain things into the cell. Literally, oftentimes, these interferons, the response of the neighboring cells is to just eliminate mRNA, even their own mRNA. They'll just degrade mRNA and they'll stop ribosomes from making proteins because they know that there's a virus in the vicinity. And so they're not going to take any risk, and they're just going to eliminate mRNA, and they're going to prevent protein production. And so that's actually going to slow down the cell's actual normal metabolic functions, but it's going to protect them from being, or potentially protect them from being infected by a virus. So interferons are a really cool example of some of these support proteins within our body. Then an even kind of bigger system of proteins that are supporting our immune system are called the complement system, or the complement proteins. And these, they're called complement because they complement our immune system. These are 9 different proteins that are in our blood, and they tend to be active by being broken down. That's kind of their mechanism of action, is when they have some sort of signal in their environment. We'll talk a little bit about some of the different signals or triggers. Then they actually start to work together to initiate an immune response and to target an invader and eliminate it. Okay. And so essentially, the complement system, these 9 different proteins, they work together to call out. They call out to phagocytic white blood cells, and they activate mast and basal cells, those ones that release different types of chemical alarms and cytokines. They also specifically actually aid in tagging a pathogen so it can be phagocytosed. And they make what's called a membrane attack complex, which literally pokes a hole inside of that invader and leads to apoptosis of that cell. They also promote fever and inflammation by activating the immune system, both directly and indirectly. Okay. And so the complement proteins can be activated in a number of different ways. One, through antibodies, and that's part of the adaptive immune system. But also, they can be activated through signals on the surface of a pathogen that seem different from the host, so non-self. Particularly, there's a sugar called mannose that tends to be on various types of bacteria, and it's not on the surface of host cells. And sometimes that will lead to the activation of what's called the lectin pathway. So the classical pathway is antibodies. The lectin pathway is mannose, or just differences in the cell. And then there's an alternative pathway, which is just sort of random breakage of these proteins. If they break and then they associate to a pathogen, if that pathogen is different from the host, then it might actually hold on to that protein for longer than a normal host cell might, and that would lead to the alternate pathway. Now, you don't need to know how all of the different proteins work and break apart and reassociate, but know that either because of an antibody or the presence of a pathogen, then these proteins will start to break apart and reassociate in a way that tags the pathogen so that a phagocytic white blood cell can take it out, calls the phagocytic white blood cells to come to the area of the infection, and also make these attack complexes that actually break apart the invader themselves. So There is a video in the description called Complement System. Go ahead and pause this video and watch the complement system video, and then come back into lecture. Okay, folks, welcome back. So, our final part of chapter 15 is on inflammation and fever. So, a lot of the different chemical signaling that we have talked about, and we will continue to talk about because there's more to say, that are happening within the immune system when a pathogen is recognized, lead to inflammation and fever. And although those can be uncomfortable, they have a really important purpose. So inflammation, it because of vasodilation, where you're dilating your blood vessels and releasing more blood and more white blood cells and more resources into an area, platelets, for example, so that we can cover and clot an abrasion. And all of that is really important to leading to taking out any pathogens, clotting and covering up any abrasions, and also recovering and repairing any tissue and cell damage. Now, that's all really important, but chronic inflammation, so acute inflammation is really good. Chronic inflammation, where there continue to be signals for inflammation from that area that don't abate, that don't stop, that's when we can actually have tissue damage because there's too much going on in that area and it's not really able to heal. Okay. Um, and so inflammation is really important. And inflammation is actually triggered by a number of different chemicals that are released by our cells. So we have our bradykinins, prostaglandins, leukotrienes, and histamines. And these are basically released when we have damage to our blood vessels. Or we have some sort of alarm that comes, for example, from a basophil cell. So what that means is damage to blood vessels. That means that we have an abrasion somewhere in our skin. If we have an alarm from a basophil or a mast cell, that means that there's an invasion of a pathogen. And so either of those will lead to these chemicals being released that cause inflammation. And that allows us to deliver to the site of injury the resources that we need. Okay. Another thing that happens as a response to some of these chemicals um, is fever. And fever is basically our body regulates our temperature at a very constant state. That's really important to our body's function and to maintain homeostasis. But sometimes, as a response to pathogen invasion, an infection, we get an increased body temperature. And that is because all of these different chemicals, they trigger a response in the hypothalamus that leads to an increase in our body temperature. Now, we don't quite understand all of what's going on, but we know that there are a number of chemicals that will lead to fever, and we call these pyrogens. And these could be bacterial toxins, they could be different types of contents released from bacteria that are being degraded by our immune system. It could be antibodies within our antibody antigen complexes, so the recognition of an antibody, or other different types of chemicals that are released as our white blood cells break down invaders. And so the exact mechanism is not clear, but what is clear is that there's an increase of temperature. And it seems that that increase of temperature is associated with increased metabolic activity, increased movement of these white blood cells throughout our body so they can get to the site of infection, and the ability, increased ability of those white blood cells to do their job. And in that way, heal and take out the infection and heal the body through recovering cell tissue damage, so on and so forth. And so, fever, although uncomfortable, can actually be a really important part of our immune system response. And In our final video, if you go into the description, there's a video on fever that gives you some information about fever and what we know about it, although we don't know everything about fever. So that's essentially our introduction into the immune system with chapter 15. And we will continue our discussion of the immune system with chapter 16. And we will continue the discussion of how we defend ourselves against invaders as we move through 16, 10, and 17. Um, Remember to go through, finish up your discussion post that talks about how skin, sweat, blood, tears, and pus and snot protect us as an initial barrier against infection. And I will see you in the next lecture. Take care, folks.